Hi everyone, welcome to my channel Pure Biology. Today I come with another video, that is the fate of the receptors, and this is the topic related to obviously endocytosis. That means what happened to the receptors and the ligands just after the endocytosis or exocytosis. Okay, so before going to this topic, I want to say that if you share and subscribe my channel, then obviously hit the bell icon for getting the notifications of any uploading video. Okay, so here let's continue the fate of receptors. The four situation may be happen to the receptors and the ligand. So first case, that is the receptor recycled and the ligand degraded, and here I have taken one example, that is the LDL receptor. Okay, this LDL receptor here, they have the low density and these are lipoproteins. Okay, the ligands, the LDL receptors carrying the ligands, these are very low, ligands have very low density, and obviously these are the lipoproteins. So first LDL that binds with the receptor, and the LDL is released from its receptor in the endosome, and the receptor recycles to the surface to be used again, and LDL is sent on to the lysosome where it is degraded. So here actually the ligand is LDL, okay, and the receptor is the LDL receptor. So this endocytosis is possible when the LDL receptor that binds with the ligands, and these ligands are the LDL, these are the lipoproteins and they have very low density. Okay, so what happened to this ligand? Actually this receptor that release the ligands, and these ligands enter into the lysosome for the degradation. Okay, but the receptor, that what happened to the receptor? This recycles again, okay, to the surface for again continuing the endocytosis process. Okay, so what is saying that here the receptor recycled and the ligand is degraded within the lysosome. The second pathway here, the both are recycled, ligand and the receptor both are recycled. So here example I have taken, that is the transferrin receptor. Okay, this provides a classic example of this pathway. So transferrin receptor that is bound to the transferrin carrying iron. Here iron is the ligand. Okay, and this iron releases that in the endosome, and the receptor now bound to apotransferrin and recycles to the surface where the receptor and ligand dissociate. Okay, so what is saying that here in case of transferrin receptor, the receptor bound to the transferrin carrying iron, and this releases that in the endosome, within endosome it releases, and receptor now bound to the apotransferrin. First it was bound to the transferrin, now it was bound to the apotransferrin where iron is absent. Okay, and recycles to the surface where the receptor and ligand dissociate. Okay, I think I thought that iron is the ligand, but here the iron is not ligand. Clear? Iron is not ligand. Iron is the molecule that is bound to the transferrin receptor, that is already bound with the ligand. Clear? So here what is happening, that iron released within the endosome, and this transferrin receptor that is bound to the apotransferrin where the actually iron is absent. Okay, and in that situation the transferrin receptor and the ligand both go to the cell surface. Okay, and their receptor and ligand both dissociate from each other. Clear? So I told you that iron is here ligand, but no, iron is not ligand. Ligand is another molecule. here the actually endocytosis is occurring with the iron molecules. Simple word, in simple word. Third one is the receptor and ligand both are degraded. So here what is saying that the epidermal growth factor receptor binds its ligand and internalizes it. Although EGF means epidermal growth factor and its receptor appear to dissociate at low pH, and they are both carried on to the lysosome where they are degraded. So here receptor and ligand both degraded within the lysosome. So what is happening here? That epidermal growth factor receptor that binds its ligand and internalizes it, not dissociate ligand from the receptor. Ligand that already have bound with the receptor totally internalizes, okay, and both receptor and ligand go to the where the lysosome for degradation, okay, and this is happen when at low pH. Number 4, that is the transcytosis. What is happening in the transcytosis? It is a process of transcellular transport which combines both endocytosis and exocytosis, and it is employed to import an extracellular ligand from one side of a cell, transport it across the cytoplasm and secrete it from the plasma membrane at the opposite side. What is saying that? That here the exocytosis and endocytosis both possible in this process, okay, and here what is happening? Import of extracellular ligand from one side of the cell. That means this is cell, the ligand is carried from this side of the cell and that across through the cytoplasm and released from this side of the cell. Clear? So here this is happening. And example is that movement of maternal antibodies and that across the intestinal epithelial cells of the newborn rat. This is the example. Okay. So actually here the fate of receptors I have already explained here. So I think you all totally understood about this topic, the fate of the receptors. Okay. So generally 4 pathways possible for the receptors and the ligands fate. Okay. First one is the receptor recycle and ligand degraded. Second one is the receptor ligand both recycle. Third one is the receptor and ligand both are degraded, and fourth one is the transcytosis. Okay, so in the next video I will try to explain about the exocytosis. Okay, this is another function of the plasma membrane. Okay, so thank you.